I advertised this as a talk about DNA, which I'm going to not go into in too much detail, just because I thought it probably too complicated for a 15-minute talk. So I'm going to talk a little bit about some historical background and as a context to our modern day work. So when I tell people that I work on TB who aren't in the field, one of the most common responses I get is to say, TB, oh, that's on its way back, isn't it? Because people have an idea that it disappeared. But they do have a memory that there was quite a lot of TB in this country at least, and certainly across Europe and North America as well, in the early and mid parts of the 20th century. And some of the older generation will remember initiatives like this, so mobile mass X-ray, where vans would come into communities and do active screening through X-rays of individuals to look for TB. This was sort of phased out eventually in this country by I think it hung around till about the 1970s when they were picking up more lung cancer than TB, and it wasn't really an effective way of controlling TB. Of course, the rates of TB in Western Europe and North America had been plummeting long before they had bans, and this is a fairly well-known trend. You may have seen this in lectures previously, but the peak of TB in this country was around about 1750, when it was responsible for almost a quarter of all deaths in the country. And then the rates fell steadily over the next few hundred years. And you can see plotted on that graph the moment when TB itself was identified, when BCG, the vaccine, was rolled out, when the first antibiotic was introduced. And those are just markers on a trend that was already falling. And probably one of the reasons it was falling was because of increased affluence, less overcrowding, better diet, and so on and so forth. Of course, this idea that TB had gone away wasn't true. It might have gone away from here, but there was plenty of it elsewhere in the world. And if you look at a modern-day TB map now, the darker green areas are those that have most of the world's TB, and the lighter green areas are the ones that used to have a lot of the TB. If you go into the historical literature to try and find out the historical patterns of TB, however, it looks like most TB was genuinely in Europeans, and there was little of it in some of those areas that are darker green on the map. Now, I'm not going to read through this. You can probably see this, but the gist of this is that this is very sort of old-fashioned language written by a British military doctor in 1914. But he's basically saying that in India, where the British were the colonial rulers, the TB was predominant amongst the British soldiers. And it was in the later half of the 19th century that TB became more prevalent amongst Indians and less prevalent amongst the British troops. And he also says a similar sort of pattern was seen in Sudan, again, where British troops were there, and it seemed like locals had had less exposure to the disease. So if you want to superimpose the sort of epidemic curves in Asia and Africa on the European trend, then I've just, in a very non-scientific way, drawn a red line there, but roughly this is what we're seeing. We see a decline in TB in Western Europe, giving people the idea that this is a disease that's disappearing, whilst there's an increase in rates elsewhere. And a colleague of ours in Switzerland called Sebastian Gagneux has done a lot of work on the genetics of TB to try and understand the historical patterns. And the top map is really a map of TB that's spread throughout the world with the migration of humanity. So humans evolved in Africa and then left, and or some of them left, others went west, others went north, others went east. And you can see with all the different colors, those indicate different genetic lineages of TB. So these days we see a lot of a different genetic type of TB in East Asia than we tend to see in Europe. But if you look at the lower graph, this is the explanation, probably the explanation for why we see a lot of European lineage TB in other countries, is that whilst the rates were very high in the 18th and 19th century in Europe, Europeans were busy colonizing the rest of the world and spreading that lineage of TB around to the other countries again. So even though it had originated from Africa, it was reintroduced into Africa and into Asia and South America and North America through these military expeditions. How was it spread? Well, nobody really understood this in the 19th century. And this was an eminent physician giving a lecture in Boston in 1864. And the lecture is entitled, "Is Consumption Ever Contagious or Communicated by One Person to Another in Any Manner?" And you can see the opening sentence here. This question may seem of very little importance because we all may be of one opinion in favor of the negative. It's an absurd proposition that consumption is at all infectious. This changed in 1882 when Robert Koch identified the bug, the tubercle bacillus that causes TB. It satisfied these postulates of his. So the idea was that if a bacterium is always found in conjunction with disease in an animal, and it's never found in the absence of disease, and if you take that bacterium and put it into another animal and it causes disease, then that's proof that this is the cause of disease. And he killed lots of guinea pigs in the process to prove this. Pretty much since then, we've been growing TB in culture, and you can see pictures here of solid agar, a thing called Loewenstein-Jensen slope on this side of the screen. So the green stuff is the agar, the yellow stuff is the TB growing on it, or liquid culture, which is what you see on the far side there, those breadcrumbs of the TB growing in clumps. We diagnose TB having cultured it. We try and culture it in the context of antibiotics. So if you look at those two tubes down the bottom, you might put antibiotic in one and not the other, and then you race them to see which one grows faster. And if it grows slower in the context of antibiotic, then that suggests the antibiotic's doing something, it's inhibiting it. So that's sort of the basis of drug susceptibility and drug resistance testing, and has been ever since drugs were introduced in the 1940s, up until the last few years. And now we're starting to move into this digital era where we're starting to sequence the genomes and look at the DNA of the bugs and derive the information we want to treat patients from that DNA. And these are some of the machines that we're using. So this big machine here on the left is one that we use a lot, but actually you can see a hand poking out of the side of the picture there, pointing at a little thing that's connected to the laptop, and that's one of the more recent sequences. It's so small we lost it the other day. I couldn't find it. I was looking in drawers, the size of a mobile phone. In the corner here, and this is getting slightly ridiculous, this one actually plugs into an iPhone. We're not using this yet, but this is the way, at least not this tiny one here, this is the way the technology is going. So you've got portable technology that's able to sequence all the DNA from the TB. In order to make use of that, we are trying to collect TB strains from all around the world. So this is a project we're heavily involved in and coordinating, and we've got partners right across the globe, and from each of those places where you see a red spot, we're collecting TB samples. And we're trying to collect up to 100,000 TB samples. We're sequencing them all. We're looking at the DNA. We're comparing that to the results from culture to see whether they're susceptible to drugs or not. And then we're learning about patterns of DNA such that we can read the DNA in the future and make predictions that tell us this drug will work, this one won't, and therefore this patient will best benefit from cocktail B or cocktail A of drugs. So that's the direction in which we're moving. We're already, I promise this is the only graph or data that I'm going to show. We're already at a point where our understanding of the genetics is so good that we can give drugs on the basis of the genetic information when we have simple drug-sensitive TB. And the point of this graph is just to show that all the colorful lines in the top left of the graph show the accuracy of our predictions. So more than 95% of the time we are, or more than 98% of the time, we're correct when we say, on the basis of the genetics, that these drugs will work, as long as the prevalence of resistance is under 40%. And that's true pretty much all across the world still. So this is telling us that the genetics works for first-line treatment. We need to learn a little bit more about drug-resistant TB to be just as accurate for second and third-line treatments. The other thing we can learn from the DNA is about transmission. So if you have two genomes, you sequence them, one on the left, one on the right, and you compare them, and you count the different mutations across those. So if you hadn't spotted it, There's one mutation between those two. Bear in mind, these are 4.5 million letters long. So this is just a snapshot. What does that mean? Just like if you have a sibling or a parent, your genetics will be much closer to that family member than they will be to a stranger in the room, presumably, unless there's something funny going on. In the same way, you can do that for TB bugs. So here's a colorful picture. Every blob is a TB bug, and the lines with the numbers on them indicate the number of mutations between bugs. So you can see in the far corner there, you've got a collection of blobs that are very closely related, and that suggests that each of those TB bugs are family members, if you like. So they will have been transmitted in the recent past between individuals who were infected with those, whereas the others are quite a few mutations apart and probably aren't family members, are quite distant, they're unrelated. Now, as it happens, all of those strains came from some refugees who made the perilous journey from Libya to Western Europe in the last few years. You'd have seen on the news people traveling across in these overcrowded boats in horrendous conditions. And a small number of them were infected with drug-resistant TB. And they were diagnosed in all sorts of different countries, Switzerland, Germany, France. So they ended up in different places, but when we sequenced the genomes, we found out that they were all very similar. So there were those in the far left-hand corner that were all of the same family. And yet these patients came from right across the Horn of Africa. We're looking at 4 different countries, large countries. So what was going on? So we looked into this a little bit more deeply, and we saw that actually most of the people who had very similar genomes had been through one single detainment camp in Libya. And if you look at, so down this, I did promise there was only one graph, this is the second. But if you look down the y-axis, you'll see annotations that mean absolutely nothing to you, but each of one of those indicates patient. And the time goes that way. So you can see the colorful bars that indicate which country each patient was in at which point in time. And the only key thing to look is horizontally look down and find all the green blobs. And you'll see, that's the time at which each patient, we know each patient spent some time in this very particular detainment camp where people smugglers kept them in horrendous conditions in Libya. And our theory is that that is where TB was transmitted from at least one individual to all the others. So the genetics gave us the hypothesis. The epidemiological investigation allowed us to come up with an understanding of how it was transmitted. And although this is from Google, this is exactly the sort of environment that was described by these people in which they were detained. Often no windows, hugely cramped, no room to move your arms and legs, perfect conditions for spread of TB. You don't need many sick people there to infect others. So we've been using the genetics of TB both to understand drug resistance and to understand transmission. And this country became the first in the world last year to implement routine sequencing of TB genomes into diagnostic care. So every patient in this country now gets their TB genome sequenced and diagnosed by this method, rather than by the old culture-based method that we showed you. So this is all very nice and good, but are we going to help stamp out the disease this way? So the WHO has set us a target saying by 2035 we want to reduce TB down to negligible rates. And to achieve this, we need to manage enormous reductions far faster than we are currently achieving. So what's the role of our slick new technology in ending disease? I'm just going to finish off with a very, very simple model, but hopefully it highlights the problem. So you have a sick person with TB who can cough on another person who might initially be infected with dormant or latent TB. So they're infected, but they don't have any symptoms. Because they don't have any symptoms, they don't spread it on, and so the next person they encounter is uninfected. So that's our little chain. What can you do about the sick? You can isolate them so they don't meet anyone else and don't spread it. You can treat them, and you can diagnose them to start with. Those with dormant TB, you need to diagnose them. You need to figure out who, without symptoms, has TB and how to treat them. And you can vaccinate the uninfected. The numbers are staggering, though. There are 10 million who are sick every year. There are 2 billion, that's a third of the world's population, who have latent TB. So how are you going to find those and treat them? And then if you take a step back, well, even just dealing with the sick ones is tricky. You can isolate, treat, but treatment is often toxic. Isolation is difficult when there are lots of people and few resources. Diagnoses are often unavailable. So although we've got shiny new machines here, they're not available throughout the rest of the world necessarily. If you're going to try and treat someone with dormant TB, good luck trying to convince somebody who feels utterly well to take tablets that make them feel horrible for 3 months or 6 months. And diagnosing them at the moment, there's a real problem with specificity. So if we do the test, it doesn't and it says they've got the disease, it's not necessarily true. I'm going to sum up the field of vaccination with one word: it's difficult. So where's our role here? We need people from all different disciplines. We need vaccinologists, sociologists, economists, chemists looking at drugs, immunologists thinking about the dormant TB, and our little role, the role for sequencing, our job really falls into the bottom left-hand corner here, diagnosis. So I think we can contribute to the eradication of TB. We don't have the solution. This is going to be a multi-pronged approach. Lots of people in our consortium, as you can tell from all those places we're gathering data from. But thanks.